So now we are going to discuss with respect to next database, which is called as a Prosite database. And this particular Prosite database will be available on the xpc.org. Okay, so we are going to see what is Prosite now. Okay, so it is a Prosite. It is a database of routine domains, family, and you will find the functional sites with respect to your search. So this particular Prosite you will find specific for COVID-19 of SARS-CoV-2. Okay. Apart from this, you will find that there's how to search in this particular Prosite database. So you can search by the IDs, okay, or you can search directly by the name of the protein from here. And different browse options are available based on the search. You can perform the browse, and then a simple search is nothing but taking a faster sequence or taking a Uniprot KB database accession number from here, or else you can also include the PDB ID from here. Okay, so we are going to use example. Okay, so example is nothing but we have used the Uniprot KB ID from here, and you have to simply click on scan. Okay, so here by clicking on the scan, you will get the results with respect to your search. So remember, the search you have been given over here is nothing but the Uniprot KB. Okay, and after the search is done, you will see that is nothing but the scan Prosite results viewer. It's available over here. Okay, so output is nothing but you will find here. And now we are going to see what actually we are getting once we click Prosite database. Okay, so hits of Prosite release in 2021-03. Okay, and it contain a motifs of the sequence which respective protein of the Uniprot KB, and this was stopped. And then you will get that found 13 hits in one sequence. So what is exactly it's about? We are going to see. It's nothing but the Uniprot KB accession number, and it is a gene which is a name of ANPK with respect to human, and they have given the amino acid sequence. Okay, and they have given the name of this full name and the EC name, and again the full name has been provided. This is how the full information has been given over here. Even you will get the chain information. So this is nothing but the sequence, and remember this sequence is about 1019 amino acids. Okay, so here you will find that the legends are given. So what is this legends is about? Okay, which contain the disulfide bridge, active site, other ranges, and other sites. So we are going to see where exactly this legends is been given as a highlighted things. Okay, so here is nothing but you will find that a graphical representation of domains which is displayed hereafter. Okay, so this is nothing but that colors and shapes of including the homology of and the structure of the functions. So this you can find that is the heads of profiles what we got. Okay, so we are getting the domains from here. These are nothing but the protein, and can you see this? The legends. It's nothing but this nothing but the disulfide bridge, and then you will see whenever I will just select the sequence from here, you will see that it has been highlighted over here. Okay, now you can see that I have selecting the active site. Okay, because here it is given that legends is about the active site. So these are nothing but the active sites of the in the protein. So whenever you are going ahead for molecular docking study, this is the most important thing what you need. Okay, because we have to know which is the active site in our particular protein's 3D structure. Okay, so this will help us to perform the molecular docking at that particular titration or the active site. Then we have the next they have given. So like this, they have not only given the sequence coverage with domain active site, but also they are giving the scores with respect to that. Okay, and the positions for the same, and which are the amino acids which has been included in that. Okay, they are providing the motifs and also the domain sites. So when you click on that, the page will redirect towards the domain database. Okay, this is how the scores has been given, and then you will find that underline is nothing but the domain part. Again, the disulfide, the domains. If you click over here as a site, you will find the site. Okay, it's nothing but the active site. This is how you will get with respect to the domains, active site, disulfide bridge with respect to the given input sequence. Okay, so here you are getting the function as well as the particular domain information with respect to your search. Okay, this is how you can utilize with respect to the Prosite database. Okay, so to again go with respect to back. Okay, so we have the, even you can give maximum 10 sequences from here. Okay, to get to know the particular active sites in the region. And then this is regarding the Prosite database. So with respect to output, we got the domains, okay, family, and also the functional sites in our given search. So this is regarding the Prosite database. Even you will find the documents over here to have a manual to go to how to perform the tutorials with respect to that. And then you can even download from here. This is nothing but you will find in the downloads. And this is about the Prosite database. Okay, so now the next database what we are going to see is about the homology modeling. Okay, so homology modeling you will find in the Swiss prod database. Okay, so in Swiss prod database you will find, or else you can go towards the Exposé server. And Exposé server is simply like NCVI database where you can find a different database with respect to your search. So here I will going to type as a homology modeling. Okay. Once I click on homology modeling, you will get this particular link, that is homology model repository for protein structure modeling. And once you click that, you have to click on browse the website. Then you can go towards the so now we are looking towards the demonstration with respect to the Swiss model server. Okay, so what is Swiss model? Again, you will get in this Exposé server. Swiss model is nothing but a fully automated protein structure modeling. Okay, and it is available in the Exposé web server. So you have to simply click on start modeling. Okay, whereas you can also look towards the specific species of your interest. Okay, again for SARS-CoV, they have given a separate work to be related to find the 3D structure. Whereas this is what news regarding the Swiss model service has been given over here. So we have to simply click on start modeling. And once you do that, again, the same thing will be available. That is, we have to give the FASTA sequence. Okay, so the search will be, that is, the format should be a FASTA, or you can give the cluster. Okay, or you can again simply use the UniProt ID accession number from here. And now we are going to add a sequence if the sequence is available from us. Okay, or we can even give a accession number of the UniProt ID. So in this particular search, we can give the UniProt accession number or the FASTA sequence. For that, you can go towards the UniProt KB database, okay, and simply click on the UniProt ID. So this particular ID can be given in the search, or you can go towards the FASTA sequence. Anything will be possible. So we'll just copy this UniProt ID and paste over here. Okay. So once you do that, you will see that the targets is started to find. So how it has been done is nothing but the sequence will be iterate, okay, in the database and try to find the particular models with respect to the given search. Okay, so here is the pop-up which has been given with respect to the search what we have given over here. And you have to simply the title has been provided as a default, or you can just reset your title based on your particular target of your interest. Okay, you can give the email ID, it will be an option. Okay, and you have to just click on build model, because search template will be done automatically, because based on template only the model will build. Okay, Now we are going to click on build model. Once you do that, it will take certain minutes, like 5 to 10 minutes, based on what is the length of your sequence. Okay, and here the sequence definitely will be a protein sequence. Okay, and based on the sequence length, it will start building the model and start searching the templates. Okay, so only based on the templates the model will generate. So how many models we will get is nothing but the 3 best model what we get based on the sequence what we have been provided and based on the templates which has been generated. Okay, so it might take another 2 to 3 minutes to generate the sequence. Okay, based on our target what we have been given. So we will wait for 1 minute. Now you can see over here that is based on our particular given search, the model is been generated. Okay, so now we got one model over here based on the 50 templates. Okay, so it will be default 3 best model will be created for your search. Okay, and here you will see that is what is exactly the templates is about. Okay, so templates are nothing but the sequence which has been taken and also the PDB. So we are going to see the next what is PDB database. So based on the PDB only, the sequence has been a page, it has iterated, and then it has given some scores, that is GMQE, QSEQ, identity, okay, method, and then we have oligosteo and the ligands. So based on that, this only you have found the particular 3D structure has been generated as a form of model. Then sequence similarity you will find over here. Okay, you see, yes. Then we have alignment. Okay, and more you will find different template search with respect to the logs. And then what actually we got the model is nothing but this. This is the 3D structure what we got, and this is nothing but the ligand which has found over here. Okay, it's nothing but the active site what we saw in the Prosite database. This is the other thing, that is ligands monomer, and then you will find whatever the informations which are available over here. And then we have the scores, which is then the estimation of the quality, and then different scores which is available over here, and the templates and how much identity it is. Okay, 100%. And then you can just have similarity and the coverage or information you will get over here. Project data is available, you can download in the zip, or else you can download this particular model in PDB format, MMCI format, recent format, PDB format. Different formats are available. Okay, so this is how you can create your 3D model, okay, of a given sequence using Swiss model server. This is as simple as this using this Swiss model server. You will also get a template alignment, okay, and use the templates for alignment. So this is how you can do, but as a beginner you can just go with respect to pasting a sequence or just giving the particular Unicode accession number and start to build the model based on the templates. This is called as homology modeling by using Swiss model server. Now we have moved towards the RCSB PDB database, and this is the most important database is regarding the 3-dimensional structure of the protein, DNA or RNA you will find over here. Okay, so you have to simply click on Google search as a PDB database and you will get this particular link. And once you click on that, you will get a home page of this. In this particular home page, you will find the how many data sets are available in PDB database. And then you will get to know from here that is how to search. You can search by simply a keyword. Okay, And based on the keyword, you will get the particular 3D structures with respect to then how many number of hits are available. Here you will find another information like molecular of the month, and then different lists are available and different publications are also available over here. You can simply search from here as a keyword. For example, TP53. Once you search on TP53, you have to click on TP53, and then you will get a different informations from here. First of all, you have to see that for TP53, how many number of hits you are getting. You are getting 273 structures. Okay, so it will be a 3-dimensional structure. So again, the filters are available with respect to organisms, taxonomical information, experimental methods like X-ray diffraction, NMR, and electron microscope. Okay, then you will find the polymer entry type. It's a protein, RNA, or DNA. Okay, then you will get the resolution based on the particular experimental methods. Then you will get the release date. Then you will get the enzyme classification name, that is heterase, transferase, lysase, oxidoreductase, isomerase. This type of information you will get from here. And the symmetry type, it's a symmetry cyclic dihedral compound. And then you will get the scope classification of the particular protein 3D structure from here. Once you do that, the filters, you can click any of the particular hit from here. Before that, you can also download the files. Options are available. Okay, over here. Can you see this? Yes. This files options are also available, or else you can simply click on this. Once you click on that, you will get the specific information of structure summary, 3D view, annotations, experimental, sequence, genome, and version information data. Okay. So before that, I will show you how to download this. You will get the FASTA file. Even you can download the PDB, and different file options are available over here. Then you can also click on display FASTA sequence, PDB, and other options are available. This is nothing but the 4-letter code, okay, of PDB ID, what we have searched for. Then you will get the title from here, DUI. DUI is nothing but you will get the DUI of the particular paper when it get published. Then again the classification, organism, expression, mutation. Okay, when it is the data has been deposited, author name from here, and the experimental data. That is what methods have been used. Resolution, R value, R free value, and observed value for R. This you will get with respect to www PDB validations. Okay, based on that only the 3D structures has been validated. Then you'll get the literature, okay, over here. And then you will find the how many chains are there. So we have only one chain in our particular protein structure. And then you will find the particular information, that is the sequence length, organism, and the image. Okay, you will also get to know the particular protein feature view based on the reference sequence. Okay, that is a PFAM information. Okay, whatever we saw in the previous video. Then you will get to know from here is nothing but the small molecule. If the drug has been bind, so which is the molecule which has been taken to bind. So you can click on the drug by clicking the structure. Okay, or else you can simply click on ligand interaction to get to know the interaction of the particular drug with respect to the protein. Okay, once you click that, you will get the next page to get reopened, that is a 3D view. Here you will find the particular 3D view. okay, of the particular drug which is interacting with protein. And from here again you can make the different visualizations. Okay, you can hide the cartoon view for the ball stick, anything you can do from here. And then we have the particular drugs. Whenever you select, it will show you the chain information and also the amino acids which are interacting, and then you will get the particular drug information from here. Okay, so here you will find that whenever you just select the interacting, so dot is nothing but the interacting residues. So whenever I just go towards the residues, you can see that the residues are highlighting from here in our sequence. This is how you can visualize the 3D view. But once we go back, and then you will find that okay, so this is nothing but the experimental data and the validations, how the experimental data has been extracted and how it is done the validations. So which software is being used for validations, that information has been shared over here, and you will get the entry history information. Then after this you will get annotations. Okay, so in annotations you will get based on the CAP database or the annotation system, protein family annotations, gene product annotations. So this is the information what you get in X annotations, whereas in experimental you will get that the data which is extracted, that is nothing but the 3D structure in the form of X-ray diffraction method, the crystal property you will get, and then this is how you will get the different properties with respect to the structure, that is crystal data information, and then you will get the diffraction information over here, and then radiation source, how the data has been collected. Okay, if it's collected from the sample, how it is done, the steps has been written over here. Then the structure refinement is very important step over here, how we are defining the structure once the structure has been extracted from the sample. So based on that only the R value, R free value has been given, and then the resolution has been defined based on the refinement only, and which other softwares has been used for the validation. This overall information you will get in the experimental tab, whereas in the sequence you will get the particular sequence of the 3D structure. So if I click on the sequence, okay, so you will get the particular sequence, but here you will get the particular chain, that is okay, and then you will get the view in 3D you can do, and the PDB instance is nothing but the chain, Uniprot ID, secondary structure, and then the Ramachandran plot. All this information you will get. So if we click on the particular colors which are indicating, you will get what exactly this is defining with respect to the plot. Okay, then the sites, cath, EPAM information, binding site, all you will get from here. Okay, you can also download this file. Okay, and also display the file in the FASTA sequence. So if I click on FASTA sequence, you will again get the same thing, that is a FASTA sequence from here. Now we are going to click on genome. Once you click on the genome, so definitely you will get the particular genome viewer from here. Once we click on genome tab, you will get the exact, that is a chromosome location. Okay, and then the PDB entry, the same thing you will get. Okay, that is on the chromosome number 12 position. All has been given over here, which is the reference sequence. Genome has been taken, that is a 38. That overall information you will get, and here is nothing but the version, okay, which version has been used for this particular 3D structure development. This is how you will get with respect to PDB ID. So this is again you will get, that is how many atom counts are there, total structure, width,
and then deposited residue unique protein chain. So we saw that is only one chain, that is a chain is available. This is how you can just take the help of PDB database to get to know the experimental data analysis and also the particular 3D information with respect to given search in the PDB database.